let's see, it looks like we're pretty close to the number of people we had watching earlier on. So we're going to give this a go here. And bear with us. These are challenging times here as we figure out some of these things. We even did a little tech rehearsal and we kind of had things working. But anyways, it looks like you guys should be able to see my presentation and hear my voice. So I'm kind of disappointed because I actually shaved for the first time since the beginning of the shelter-in-place order here and cleaned up and everything. But anyways, much rather show you pictures of squid, and in particular, squid eggs. So I'm going to dive right in here and talk a little bit about the work. So the research that I'm going to present to you now, it's an element of my PhD work and some research that I've done at the Center for Coastal Studies. And before I talk about the research, I do want to thank a couple of other folks who are really heavily involved in this work. First of all, Ernie Eldridge, who's the senior author on this paper. He's a commercial fisherman out of Chatham, and I've worked with Ernie and his family for going on 15 years now, studying the biology and ecology of squid, among other things. And you'll learn a little bit more about Ernie's role in the project, his family's role in the fishery that Ernie works in. I also want to thank Katie Grolio, who's a master's student who graduated from Harvard a couple of years ago. She helped out a lot on some of the data analysis that I'm going to talk about. Then we had a whole host of volunteers and other folks who helped us in the field and in the lab. So anyway, the title of this talk, Babysitting Baby Squid. It's kind of a fun play on words here, and people have been coming in hot with the squid puns leading up to this. But in situ monitoring just means monitoring in place. So what we're doing is something relatively unique. We're studying these animals in the field, working with fishermen to do that. Working in the fish weirs that I'll talk about gives us a really unique opportunity to study these animals in place in their natural habitat as opposed to in a laboratory environment. So I'm going to try to advance the slides here, and hopefully that will be working for you guys as well. I'm keeping one eye on my phone, which is actually working much better than my computer in this context, and so I can see if my slides are advancing that way. But I have to wait a few minutes, or at least several seconds anyway. So we shall see. But anyways, longfin squid, Doryteuthis peeli, are, for many folks, are calamari. They're a species that a lot of folks like to eat. And they're also targeted by commercial fisheries in Nantucket Sound, around Cape Cod. Further offshore this time of year, squid are migrating in from the edge of the continental shelf. So they are harvested by Marine Fisheries from the mid-Atlantic all the way up to Cape Cod. And we'll talk a little bit more about where squid live and what they do. Years ago, this was a bait fishery primarily. These days, they're highly prized as food. But for a very long time, squid were just considered bait. And I know a lot of folks today who still won't eat squid. They simply rely on them as bait for bass and other species. And they're certainly a really important forage species for striped bass, for fluke, for a lot of other animals. So they're really important ecologically. But when you think of what they are in terms of what kind of animal they are, it's really important to remember that they're mollusks. So they're actually fairly closely related to snails and clams and oysters. But they're really, really smart snails, if you want to think about it that way. They're relatively short-lived. They live less than a year. So they kind of live fast and die young. They reach maturity at a length of a little bit under 4 inches, body length, not counting the arms and the tentacles. So they reach maturity and are able to spawn relatively quickly in their lives. And they spawn year-round throughout much of their range. And so these critters range from the Gulf of Mexico and all the way south to Venezuela, all the way up to Nova Scotia. But most of the spawning that we know about happens in inshore waters around places like Cape Cod. We do see egg masses offshore, particularly in winter and early spring, but they do seem to spawn throughout much of their range. We'll talk a lot more about reproduction and egg masses. But they drop their egg masses, they deposit their egg masses on the bottom, and they're fertilized by multiple males. And I've got a little bit of video of that to show you. They grow faster when it's warmer, and that'll come into play later on in this lecture. And as I mentioned, they do range from Newfoundland or Nova Scotia all the way south to Venezuela. Oh, hello, Candace from Acton. I see you've tuned in. Um, so as far as their biology and ecology, these are really, really interesting critters. I could talk to you probably for an hour just about how they change color and things like that. But today we're going to talk about reproduction. And in particular, as I mentioned, the females will deposit these massive egg clusters on the seafloor. Um, Males will go in to fertilize those eggs, so the male will approach the female as she's about to deposit those eggs and pass essentially a sperm packet or spermatophore to her as she's extruding her eggs. Um, and so those eggs will be fertilized as they're deposited into that egg mass on the seafloor. And what you can see at the top left of your screen here, that's an underwater video shot by Nick Kalianis. That was some footage from Roger Hanlon at the Marine Biological Laboratory. And what you're seeing there is from some of his research, also working in fish weirs or fish traps, which I'll tell you a little bit more about. Um, but what you have there, that's taken in about 25 feet of water in Nantucket Sound. And you have a female depositing eggs onto that egg mass, and you have males closing in, attempting to mate with her, and presumably successfully mating with her. You have the male at the top. It's kind of in a guarding position. He's probably already mated with her. And then you have another male coming in, what Roger likes to describe as a sneaker male, also attempting to mate with her as she's depositing those eggs to that egg mass. And these egg masses can be relatively large. That's a relatively small one there. I've seen them the size of trash cans all the way down to the size of my fist. And when they're deposited on the seafloor, if you look at the bottom left of your screen, this is what those egg masses look like if you put them under a magnifying lens. So these are relatively fresh, newly fertilized eggs. And they're going to develop relatively quickly once they're deposited on the seafloor. So in a laboratory, developmental time ranges from about 20 to 30 days. And you'll see in the field it's actually, in some cases, a little bit faster than that. But these embryos, these fertilized embryos, you'll find hundreds of them to thousands of them in these large egg masses on the seafloor. And they're going to develop relatively quickly. And you'll see in just a couple weeks' time, in a few weeks' time, all the way over to the bottom right of your screen, when they hatch, these larval squid, we call them paralarvae, because they're a lot different than a larval fish. If anyone's ever seen a fish larva, they have big googly eyes and little bodies. They don't really look like the adult that they're eventually going to be. But a larval squid or a squid paralarva, when it hatches, it's essentially a fully functioning little squid. In this case, it's barely the size of a grain of rice, but it is fully able to hunt. They have incredible visual acuity. And if you look at that photograph of the squid at the bottom right, its eyes are already incredibly well developed as it's hatched. That's just probably a 2-day or 3-day old hatchling from our laboratory. And you can see the light reflecting from those eye lenses there. Their eyes are polarized. They have incredibly good vision. They're able to hunt very well underwater. They're also able to change color. Squid have color-changing cells or chromatophores, and you can see that as a series of colored dots. And I'm going to try to show you some more video of squid hatchlings at the end of this presentation. Given the technical difficulties we've had today, maybe that'll work, maybe that won't, but we will give it a try. And a couple of other things I'll say about this little larval squid at the bottom right of your screen. That animal has been hunting in one of our tanks, and this was a few years ago. But if you look at the very top of the photograph of that paralarva, you can see there's kind of a pinkish mass. That's essentially its gut. It's been feeding. It's got essentially a belly full of plankton. In this case, we were feeding it zooplankton from Provincetown Harbor, and it was feeding quite well. You might also be able to see there's what looks like a little glob. I'm going to move my mouse there. I don't know if you'll be able to see it or not, depending on how large your screen is tuned in. But there's a little globule there. That's actually the ink sac. Squid are able to extrude ink as a way of escaping a predator, for example, a way to decoy a predator or to mask itself from a predator, depending on what it's doing. And even these tiny little larval squid will have that ink sac developed. So it's essentially these little squid paralarvae are fully functioning miniature squid when they hatch. As I mentioned, they only live about 1 year. So it's 1 year's time from that little tiny half a grain of rice size paralarva. In about a year's time, that animal will be um, upwards, in some cases, of 8 inches to 10 inches long. Sometimes you get even bigger tubes than that. And at that point, it will probably have spawned out already. So very short life cycle, capable of replenishing the stock very quickly, which is why the fisheries have tended to be relatively robust and sustainable over time. But again, really fascinating critter. Maybe if you have questions at the end, we can ask them over the comments, and I can try to answer some of them. But I'm going to move on and talk a little bit about the research that we do with these animals. Again, working in partnership with the fishing community, Ernie Eldridge and his family working out of Chatham. And so the way we study these animals working together in the weir fishery, so fish weirs are very large fixed traps. If you look at the screen on the bottom left, you can see this beautiful photograph by Shireen Davis. And what you're looking at there is a series of nets strung from hickory poles. That's in northeastern Nantucket Sound, where the stars are in the map to the right. And these large traps are set every spring. It's actually coming on to weir fishing season now. And they'll fish into sometime in the summer. And the way these nets work, fish or squid could actually swim through much of the net, but they perceive it as an obstacle. They move down along this net called a leader, which is a long fence that's perpendicular to the shore. And they arrive in the structure that looks like a heart at the bottom left of your screen. You can see the net strung from the poles there actually kind of looks a bit like a heart. And that's made of a finer net that the squid can't swim through. So they'll move through that heart. And there's a small opening here about 10 feet wide. Remember, this photograph is taken from an aircraft, so this is probably about 100 feet across. And they'll wind up in this bit called the bowl at the very bottom. It kind of looks like Pac-Man. And that bowl will hold the squid and anything else that the fishermen may want to retain, to bring in, or to let go. One of the cool things about the way these traps fish is you can untie some of those ropes around the edges or otherwise manipulate the net and release things that you don't want to catch, don't want to take home with you. So it's a really neat fishery in that way, and that you can let go a lot of small fish that you don't want to bring in. And one of the things that happens is when the squid go in there, a lot of times they spawn, they lay eggs in that trap. And that's kind of cool because considering that in many cases they're being harvested and brought home, they're laying eggs in there first, ensuring another generation of squid afterwards. And it also gives us a really unique opportunity to study the squid when they lay those eggs in the traps. So working with the Eldridge family, we keep logbooks. So we record how many squid are landed, how many fish are landed, anything that's let go in the traps, and we record when we start seeing squid eggs and over what period. We also collect environmental data. So every one of these traps, and this is really cool, these traps are essentially the same design that was being fished in the 17 and 1800s. And the trap design hasn't changed very much, but we're now putting modern oceanographic instruments on these traps. In this case, a relatively simple instrument shown here that just measures water temperature every 15 minutes. And this will go on through the entire fishing season. Some of the traps receive other sensors, and we'll show you what those look like. And throughout the course of a fishing season, if we have an opportunity, we'll collect an egg mass, and I'll show you how we do that. If a group of squid have deposited eggs in the trap, we'll take those eggs out. We'll put them in an enclosure. We'll take a little sample out of that, but then we'll put it in an enclosure adjacent to the weir. So here in this photograph, you can see Ernie with the very first version of what we called the incubator, get it, that we use to keep the squid eggs in and sample them over time. And that one is essentially made from stuff I had laying around the garage. The design of that has evolved significantly since then, but I just like this picture because it shows a foggy day at the weirs and Ernie inspecting the first version of the contraption I built. On top of that cage, you can see an instrument that in that case is measuring not only water temperature, but also salinity, how salty the water is, and how much oxygen is in that water, which is another really important thing. And so we'll take those egg masses and put them in the enclosure. And the way these enclosures are designed, it's going to maintain water flow around the squid eggs. The newer designs we have are better at that than that first one. And basically making sure that these are realistic ambient conditions, so very much replicating what would be in the natural environment for these squid eggs, as opposed to working in a laboratory. The other way this works is that cage structure keeps the other squid from going in and adding more eggs to those egg masses, because what we want to do is monitor one sort of clutch or cohort over time. And that's really important if we're trying to understand how water temperature, for example, affects the development of these little tiny embryonic squid. So we'll put that cage back on the bottom next to the fish weir, and we will sample it usually twice a week. And we'll sample a little more frequently, a little faster, as we think they're getting close to hatching. And I'll show you how we know that. And every time we sample it, we'll bring it to the surface. We'll take a few of those little fingers, those strands of eggs, and bring it to our lab for analysis. We do that in our wet lab in Provincetown. It's relatively easy. All you need is a dissecting microscope. And we look at the embryonic development stage. This has been classified in the laboratory by scientists at the Marine Biological Laboratory. So we look at there's a set of stages of progression of embryonic development that we can use to kind of compare across the samples that we're looking at and make sure they're all developing at the same rate, the same speed. And like I said, if we look at these egg masses and these egg samples under a microscope, and we know from experience and from this developmental chart that they're going to hatch relatively soon, we will try to go back out and sample almost immediately so we can actually capture the hatch in place. And that's really important because we want to establish when they were deposited, when those eggs were first laid, and we can do that because those traps are hauled every day, and then we want to know when they're hatching. And again, we know that from direct observation of those little larvae coming out of the egg strings, and I'll show you what that looks like in a second. So going through results here, first looking at the log books, you can see over the years of this study, Katie and I looked at data from 2008 through 2016, and what we found was that there's a pretty wide variability between years. Some years we have a relatively short period in which these eggs are seen being deposited in the traps. Other years they spawn over a much longer period. So a lot of variability from year to year. And when we first see the eggs changes from year to year, when we last see the eggs changes from year to year. You can see there's, again, a lot of variability between years. And as we collect this data over time, we'll see if this is changing over time. And we were able to sample eggs almost every year, at least a couple of egg masses each year. So when we're bringing these samples back and looking at them under the microscope, this is what that looks like. You can see a progression of embryonic development as seen under a microscope. So you can see three photographs to the right of your screen. The photograph at the top, kind of at the center, is essentially a newly deposited egg that's likely hours to maybe a day or two old. The next photograph over, kind of in the middle there, as you go towards the right of your screen, that's probably a week and a half old. I'd have to double check that. And what you're seeing is something that already looks like a tiny little squid. Its proportions are very different than an adult squid, but you can see a lot of the same features. At the very top, you can see the muscular mantle, which is the body wall of the squid. You look very, very closely, you can see something that looks like a pair of ears. Those are the tiny fins that the squid will eventually use as part of its propulsion. Big googly eyes, and then the big thing that looks kind of like a ball that it's holding in its arms is actually a yolk sac. Just like a lot of other animals, these embryos are living off of a yolk as they develop inside the egg mass. And all the way over to the right, you have an embryo that's pretty close to hatching. So this is still inside the egg mass. This is what they look like when they're inside that gelatinous capsule. Um, it's still got a yolk sac, although it's much smaller now in proportion to its body. There's another yolk sac that just happens to be next to it from another embryo. This was excised from the egg so we could get a better look at it. And you can see it's got those color changing chromatophores, those colored dots already. So even in an embryonic stage close to hatching, so this is an animal that would not hatch yet. Think of this as almost like looking at an ultrasound in a mammal as that fetus is developing inside. This is kind of the same window into development. You have an animal that is close to hatching here. It really does look like a squid. And probably within the next few days, that animal will hatch. So if we saw that under the microscope, we would go right back out probably the next day or the day after and try to get another sample so we could capture the hatch. One thing that's important is we use these stages to make sure they're all developing at the same rate because we want to make sure we're looking at the same clutch or the same cohort, that we're getting enough samples to really track development time. And this is what we try to get as a final sample. In other words, we're trying to capture the hatch. So if we've been successful, if we've sampled at the right frequency, we'll go back out and we'll get an egg mass that's actually hatching. And so if you look at the bottom left, that's what one of those egg fingers, those strings of eggs from the egg mass looks like as it's about to go under the microscope. And then in the center, you can see little newly hatched paralarvae that have come out of that string as we were watching it under the microscope. So you're actually capturing the hatch there. When we had this happening in the lab, that actually spawned, there's another unintended pun, spawned a whole other project where many of us were actually trying to raise these things with some limited success. Maybe that's another lecture for another week. None of us were supposed to be working on that project, and we all lost a lot of time on other projects trying to keep these things alive. But I guess that's another lecture for another day. But really, really cool to see these animals hatching. As you might expect, as water temperature got warmer, they developed faster. So this is looking at the average temperature during their development. And if it was warmer as they were developing in those egg masses, they moved very quickly from deposition to hatch. If it was colder, they took longer to hatch, which makes sense. That's what you might expect. In the time it took from deposition, so from when those eggs were laid on the seafloor to when those little hatchlings were appearing, ranged from about 12, so under 2 weeks, to as long as a month's time, depending on what the water temperature was. And again, happening faster when it's warmer, as you might expect. This is the first time, to our knowledge, this had been studied in the field, and it's really worth pointing out that this wouldn't have been possible without the collaboration of the fishermen, particularly Ernie and Shannon and their family. We would not have an opportunity to access these animals in this way without that collaboration. This is something you can do in a laboratory environment, but you don't have the advantage of being able to have natural conditions there. And again, lots of references here, but basically what we saw in the field fit fairly well with the laboratory work. So that's good. We feel like we're on the same page. And that also means that probably the laboratory work that was done earlier on is probably fairly representative and fairly realistic. Love to have more samples, as all scientists do. And we could use those samples, hopefully, to model development time. So if you have a temperature measurement and you have a newly deposited egg, you should be able to predict relatively quickly when they're going to hatch. But again, we're just looking at water temperature. There are other things to consider. First of all, looking at dissolved oxygen, pH in particular, as we think about things like ocean acidification and low oxygen zones in the ocean. Those are things that are going to have an adverse effect on development. Sometimes it might slow down development. Sometimes it actually might result in a malformed or otherwise, incapacitated embryo. So as we think about climate change and ocean change, those are things we'd also like to start measuring so we can come up with some in situ or field studies that are comparable with some of the laboratory work that's been done by other colleagues. And I should point out that even in our study area in Nantucket Sound, We have recorded sometimes when it's essentially anoxic, when there's so little oxygen in the water, and that can be due to an influx of algae and nutrients. That as the algae breaks down, it can suck the oxygen out of the water. That can happen for a lot of different reasons. But for example, we had some relatively slow development times. A couple of the outliers in that figure that I showed you may well have been because there was actually low oxygen, which does inhibit development, slows down the development, even though it's warm. So other things to consider here, as there always are with these kinds of projects. But that was an example of the kind of work that we can do working with fishermen, bringing scientists and fishermen to study these animals in the field. And really quickly, I'm just going to show you a couple of photographs and maybe a video of what these critters look like when they hatch. I had a wonderful intern last summer, Bailey Regal, and Bailey took some really cool photographs and videos just using her iPhone through the eyepiece of the microscope. You know, we have this fancy rig that lets us take photographs, and her footage just holding the phone to the eyepiece are actually better than anything I've ever gotten using more advanced instrumentation. So I'm going to try to show you some photographs really quickly of those newly hatched squid. And in the meantime, if you folks have questions, maybe this is a great time to start sending your questions into the comments here. Here are some photographs of newly hatched squid larvae, paralarvae, under the microscope that Bailey shot this past summer. Really amazing photographs, Bailey, and you can see the chromatophores. Some of them are excited, some of them are not here. So when you look at these larval squid, you can see little tiny dots. Those are the chromatophores that are sort of contracted. And the other ones have much bigger dots. Those are expanded. And if I can show you the videos, hopefully you'll be able to see that. Hopefully you'll be able to see those changing in real time. But again, some amazing photographs of these little newly hatched squid. I'm just going to see if I can show you a video. Yeah, we'll leave these videos up, and unfortunately, I think I can only, I don't dare try to go back to the camera to answer your questions that way, because I don't want to break the tech here. We've so far, I think, made it through this presentation without any more failures. But please do, if you have questions, please do write them into the comments here, and I'll do my best to try to answer them here for the next little while. Hopefully, you've been able to see this presentation up to this point. I probably don't have any way of knowing that right now. Oh, okay. We have a question from Kiara. Do they maintain those chromatophores into adulthood? They absolutely do. So, I didn't really get to talk about that. Maybe we'll do a future Facebook Live presentation on adult squid. But yeah, absolutely. They maintain those color-changing cells, those chromatophores, throughout their entire lives. And they use them as sort of adaptive camouflage when they're hunting. And they also use it to signal one another. You'll see different color patterns in those chromatophores depending on what the squid is doing. And Roger Hanlon down at MBL in Woods Hole has put together an ethogram looking at what those different color patterns might mean. It's really, really cool. Maybe a whole other lecture. Kevin's son Colm has written in, do these squid live in other parts of the world? Great question, Colm. These squid, this particular species only lives in the western North Atlantic, but there are a lot of other species like these squid around the world. There's a very similar species that lives off of South Africa, one that lives off of California, one that lives in the Mediterranean. So you can find relatively similar species of squid around the world with very similar life histories. Cindy says the audio and video are working well. That's great. Thank you, Cindy. And I will stick around for a little bit here and see if any more questions come through. This is only our second experience with Facebook Live in these times of diminished bandwidth and all of us working from home where we may not have the best internet connections. But hopefully this has worked out well. Oh, Amber asked a great question. How might global warming impact the maturation of these squid in the wild? Well, that's a good question. Generally, as adults and larvae, they grow faster when it's warmer. So that can have both positive and negative effects. In some ways, it changes the amount of time those little squid are exposed to predation because they're going to grow very quickly. But it also might affect the amount of energy they have available to reproduce. Squid generally do well in warming temperatures. So squid ecologically often are the winners when something like that happens. Um, but yeah, generally they're going to grow up faster when it's warmer. One of the things that that can mean, though, is it might change the time that the squid mature, when and where they reproduce, when and where they're feeding relative to where their food is. So sometimes that fast growth rate isn't always an advantage. If you hatch and suddenly you're hungry and you're out there in the wild and you've hatched at a different time of year than when your food might be available to you normally, that could be a tough situation to be in if you're a tiny little half a grain of rice sized squid. So it can have a lot of different implications. Great question. Cindy asks, what percentage of the squid from one clutch are expected to make it to an adult? Good question. We right now don't have a good number for that. The best we can do is estimate that, but right now we really don't have an estimate. It might be something we're able to model in the future. I'm actually working on a proposal that will get submitted later on today with some colleagues of mine, where we'll try to look at the effect of different conditions on hatching success, but extrapolating from the hatching success all the way up to adulthood is really, really tough to do. But we might be able to come up with some kind of proxy for reproductive capacity if we start counting eggs using sonar and then learn what we can about survival of embryos. We might be able to model that someday, but we're a ways away from getting at that now. Candice asks, is this species of squid the one fishermen catch for calamari? This is definitely one of them. This species in particular is kind of the mainstay of the Rhode Island squid fishery. There's another species called short fin squid. These are long fin squid. And short fin and long fin squid have slightly different life histories in a lot of ways, but generally the long fin squid are a little bit more prized for food than the short fin, but both make an excellent calamari. Thanks everyone for tuning in, and hopefully you'll hear from us again soon. Take care, everybody.